So the work that we shared at the ASH meeting this year had to do with characterizing p53 and mutations and their role in leukemic transformation. So, we know that the acquisition of p53 mutation seems to be an important event in progression of chronic phase to blast phase MPN. We see that p53 mutations are not common, 3 to 5% in chronic phase MPN, but close to 30% in transformed disease, which really implicates them in a causative role for this transformation. So we wanted to investigate this further. And what we did is to develop a series of mouse models to ask specific questions such as, do you require bi-allelic alteration of p53, or is mono-allelic alteration okay? And where does the leukemia arise from, and what are the pathways involved? And so from a high level, what we identified was that bi-allelic alteration of p53 is necessary and sufficient for leukemic transformation. Transformation does not occur in mice that have JAK2 and only one p53, either point mutant or loss of one p53 allele. So you really, it seems, need two events for transformation to occur. Transformation is characterized by the development of a complex karyotype, which is really very much what we see in humans as well. And so that kind of gives, I think, some sense of fidelity to the human disease. The work that we did to characterize where the disease arises from comes from the observation that these mice develop an erythroleukemia. And in fact, it's the megakaryocyte erythroid progenitor population that seems to be enhanced and expanded prior to and after transformation. And in fact, doing in some of the work that we did, we were able to identify that the leukemia initiating population seems to come from within this megakaryocyte erythroid population, which makes it distinct from a lot of other leukemias that are of myeloid origin. Drilling down into what are the mechanisms involved here, we identified through RNA sequencing and gene set enrichment that the BMP-SMAD or TGF-beta pathway seems to be highly upregulated at the time of transformation. And importantly and interestingly, we see the same thing in human erythroleukemias, which we compared directly to our mice. This BMP-SMAD pathway seems to play a functional role. If we knock down BMP2, there is restoration of normal hematopoiesis, the mice live longer, and the leukemic cells lose a competitive advantage. So to us, that credentials this pathway as playing an important role in the process of transformation. Yeah, our next steps to further investigate this are really focusing on two things. One is to understand the mechanisms that lead to deregulation of this pathway. And number two, I think importantly, how do we target this pathway? What are some things that we can do to try to pharmacologically inhibit it? And, does that have utility in the human disease? I think those are two broad questions that we're examining currently.